Good. So I want to start by introducing everyone to the Hawkins Translational Lab, led by Dr. Cynthia Hawkins. And in this lab, our whole purpose is to develop tools that will guide us and help us into diagnosing patients, determining prognosis of tumors, and helping guide therapy. With this, we've developed, as Lucy mentioned, a series of platforms and tools that are already clinically approved and in current use. And we have a few tools that I've specifically worked on over the last few years that are under development and into the track of being clinically approved and implemented. And today I'm going to focus on the liquid biopsy and the strategies that we have been developing within the liquid biopsy scope. And the main reason that we're interested in these tools is that we do believe that a liquid biopsy will soon replace surgical biopsy in a lot of the instances in patients' settings that we have. And it will also be used to add information throughout the patient's treatment journey. So I will talk a little bit about that more in depth. So I'd like to start by defining what is liquid biopsy. So liquid biopsy is the process of acquiring through minimally invasive procedures, liquid sources that we have in our body to look for specific alterations or molecules that can guide and can tell us something about a disease process. In this case, what we're looking for are alterations that would be present in a cancer cell and that we can find in easily accessible tumors without having to do a surgical biopsy. Largely used, especially in the adult world, is blood. We can access urine, cerebrospinal fluid, and saliva or stool. I'm going to concentrate a little bit on this talk in brain tumors. And for brain tumors specifically, the source and the fluid that we found to be more sensitive is the cerebrospinal fluid. And this is the fluid that bathes our brain and spine. And it's in closer proximity to the lesions that are present in the central nervous system. Within these liquids, we can find a series of molecules or analytes that can be studied. I'm today going to focus on cell-free DNA, and cell-free DNA are really small fragments of DNA that all of our cells are shedding into these fluids, and we can find them from normal cells to diseased cells such as cancer cells. And what we're interested in really is to find alterations that are present in these tumor cells and that we can find in these small pieces of tumor DNA and that are not present in our regular body cells. One of the things that we're going to touch upon is that the proportion of tumor DNA that is present, or cell-free tumor DNA that is present in these fluids, is very, very small compared to the proportion of normal cell-free DNA that is present in these fluids. So I'll touch upon some of the challenges and things that we need to think about when we're considering liquid biopsy, specifically in pediatric brain tumors. So one of the things I've already mentioned is the small proportion of this analyte, the cell-free tumor DNA that we're looking for. The other one is the choice of fluids. So for our brain tumor patients, we would at this stage look for cerebrospinal fluid versus blood, as the sensitivity is still higher in those. Another thing that we need to think about is the source and the volume. So the way we obtain cerebrospinal fluid is usually through a lumbar puncture, or if at the time of surgery or a surgical procedure where patients need an insertion of a shunt, we could obtain the fluid that way as well. Another thing that impacts the sensitivity of these assays is the site where the tumor is located. So if it's more proximal to where our cerebrospinal fluid is located, we tend to have a higher sensitivity. And that will depend as well on the amount of disease that we find and the stage of the disease. All of that will factor into us choosing what type of test we're going to do to characterize these cell-free tumor DNA. And how can we use liquid biopsy? So one of the big things that we can do is at the stage of diagnosis and surveillance. So in specific to the scenario of MMRD patients, we're able to follow patients and use these strategies for early detection and diagnosis of brain tumors. With that diagnosis and detecting molecular alterations that are pertinent to the tumor, we can sometimes find mutations that have targeted therapies, that have drugs that are specific to those molecular alterations. So we can implement personalized therapy as well. Additionally, we can use this strategy to monitor patients. So patients that are undergoing therapy, such as immunotherapy, we can monitor over time and collect these samples over time and detect the quantity of that alteration in these fluids. And what we want to see is that over time with treatment, those levels are going down. And if there's by any chance a recurrence, we pick them up going back up. Another thing that we can do is find new alterations that with the tumor evolution may appear and may help us in guiding therapy and even adding additional agents. Finally, we can detect very, very small quantities of these alterations, which may allow us to detect an early relapse and quantify minimal residual disease, permitting us to change treatments in case this does happen before imaging detects a potential recurrence. There's many, many methods and many tools that we can use to analyze these alterations. I'm going to focus today on the two that we are developing currently, which are next generation sequencing, and specifically a panel and low-pass whole genome sequencing. So when we talk about panel sequencing, what we do is we collect the cerebrospinal fluid, we extract the DNA, we make libraries for next generation sequencing. And then what we do is with the panel sequencing, we have a selection of genes that are commonly mutated, and we sequence those genes very, very deep. We look at them many, many times, and we're able to detect small point mutations and fusions in those specific genes that are in our panel. Another strategy that we use with the same libraries that we have made is we tile and we look at the whole genome at a very shallow depth, so between 1 and 3 times. And that does not allow us to look for these more point mutations and specific mutations, but it does allow us to find large deletions or gains in the genome. So these are two strategies that we can use to detect alterations that are pertinent to the tumor. And what we've done is we've validated these panels in a retrospective cohort of brain tumors and different types of brain tumors. And what I want to show you today is that for specifically high-grade gliomas in MMRD patients, the sensitivity was really high, with 83% of them being positive. So this allows us really to implement these tools in all of those stages that we talked about. And I want to finalize by presenting a few current cases where we think this strategy was useful and how we envision using this moving forward. So on a first case, this is a teenage patient that had a diagnosis of high-grade glioma with an IDH1 and P53 mutation in the tumor. We found the same mutations present in the CSF with the addition of a PIK3CA mutation and some variants of unknown significance in other MAP kinase genes. So for this patient who is currently on treatment with immunotherapy, we can potentially envision using these results to, if necessary, add additional targeted agents. Additionally, from the same sample, we were able to profile the copy number alterations that are present in this sample and compare to what are present in the tumor as well. And this can be used over time to assess the stage of the tumor, whether once it's not picked up in imaging anymore, we are able to still detect it on the liquid biopsy. Another example of a patient diagnosed with MMRD high-grade glioma, and this patient at the start of immunotherapy had mutations in FGFR3 and P53 present in the CSF, which were also present in his tumor at diagnosis, and we could also find copy number alterations in these tumor. 4 months later, he had a picture with neurological deterioration, and really the imaging wasn't successful in showing us whether this was due to toxicity to the drug or if there was progression in the lesion. So the CSF helped us confirm that there was still tumor there and mutations found, as well as the copy number alterations that were pertinent to the tumor, helping us decide how to move forward with this patient's treatment. And in this last case that I want to show you, another high-grade glioma patient with mutations that were present in the tumor confirmed in the CSF, with also alterations in the copy number profile. For this patient, we additionally are starting to implement our logic tool, which Lucy mentioned briefly in the beginning. And the logic tool can identify a score which will predict MMRD, basically. And we've used this for previously in tumor tissues, and now we're currently developing it so that we're able to detect these levels in the liquid biopsy as well. And here what I'm showing is the red dot is this patient sample compared to tumor MMRD positive controls. And here the 3 green dots are CSF samples from MMR proficient patients that are negative. So to summarize, the way we see using this tool in the MMRD patients is at diagnosis, to be able to detect these alterations, and then throughout the patient's treatment journey, monitor response to therapy, potentially help decide whether it's the time to finish therapy, and then follow up thereafter to be able to detect early recurrences. So I want to thank all of the patients, the collaborators, the Tumours lab, and the Hawkins lab, and specifically the team that works directly with this project. Thank you. And I'm open for questions.
Thank you, Liana. That was amazing. We had a few questions, but they all kind of come to the same root, which is asking, as a parent or as a family member, is liquid biopsy already being implemented in clinic as part of the surveillance protocol as a standard of care? Or is this something that parents themselves or healthcare teams or we as a consortium need to still advocate for?
So at this stage, we have started using this on a research basis, and it is quickly being transitioned. So we're at the stage of implementing and transitioning it into clinic, which means it's been validated, and now we are in the process of implementing it clinically. It is available through the consortia on a case-by-case basis, through the case discussions, and Uri and Anirban have been connecting our team with the interested physicians and how to enroll these patients basically and get this done.
I just want to add to that, if that's okay, Lucy, that this is one of the reasons to give you updates, is that Liana and Cynthia have now the ability to do some of these things, but the only way to see if it's actually helpful is to together collect this information longitudinally. So for many patients to follow over time, take blood tests and sometimes CSF, and that's we do on a patient-by-patient basis to see if it can actually predict something to happen or actually predict surveillance and other things. So we really want your help in getting this information so we can, in maybe in 1 or 2 years, say, wow, that was really helpful for surveillance, very helpful for targeted therapy, and maybe not as much to monitor response. So this is why we